All right, so now we're going to look at how bacterial transcription is regulated. All right, so let's go back to those the idea of those operons that I mentioned in the last slideshow. So remember, operons are a group of associated genes that are all under one promoter. So you can see on the right, we have gene X, Y, and Z all under the same promoter. So they're going to get transcribed together and translated separately into 3 different proteins. The genes in an operon excuse me, are usually associated, so they make proteins that have functions that work together. And then the operons also have the operator, right, that comes it's downstream, so after the promoter, but upstream before the genes. Here, so right in the middle. And the key is that this operator is where a repressor protein can bind. And when a repressor binds to the operator like this, it inhibits the activity of RNA polymerase because it's literally physically blocking the genes from being transcribed by RNA polymerase. So this will be the key idea that we'll look at in a second. And this term, so operons create polycistronic mRNA. This means that this single mRNA strand here contains the information for multiple proteins, polycistronic. And then if it contained the information for one protein, it'd be monocistronic. All right, so let's look at some basics of operons. So you don't have to know what all this is on the right. It's just to show you that operons they function essentially like circuits. So they can regulate themselves. So the operon can regulate its own transcription, and it's like this whole very, very complex process. And you can model it using many mathematical methods. So it's actually pretty cool once you get into it. But so this is a way for bacteria to basically control when they're expressing certain genes, but depending on the environment that they're in. Okay, so let's do a brief introduction of the lac operon. So the lac operon is in E. coli, and it contains 3 genes that are essential for the breakdown of lactose. So remember, lactose is just a sugar, and we usually think about it being present in milk, but in the environment, just like when E. coli are swimming around in their environment, like lactose can be encountered pretty commonly. So yeah, so let's look at what each gene does. So, didn't mean to do that. So so LAC-Z, this first gene here, LAC-Z, codes for an enzyme called beta-galactosidase. And this is the enzyme that literally, this is the one that actually breaks down the lactose. So you have this lactose here. You don't have to know this, but it's a dimer. It's a disaccharide with glucose and galactose. And what a beta-galactosidase does, it literally cuts this bond and breaks it down here. Okay, then forgot to underline this. We have LAC-Y, the second gene here. It codes for lactose permease. So this is a membrane protein that'll help transport lactose into the cell. So lac permease looks like this, and then it'll allow the lactose to enter the cell like that. Then LAC-A, this final protein, codes for what's called transacetylase. And this enzyme actually doesn't, I forgot to finish the sentence, but it has a kind of mysterious role. We know it's essential, but we don't know exactly what it does right now. There's some speculation that it helps detoxify when the cell encounters like toxic things, but not really certain about that. So don't worry about this one. All right, so and then elsewhere in the DNA, this isn't within the operon, but we also have a separate gene. So let's say somewhere else we have this gene here of LAC-I elsewhere. And this is also related to the operon, but not directly physically inside the operon. And this gene codes for this repressor protein that we talked about, the repressor that can bind to the operator here to inhibit transcription of the lac genes. So for our purposes, we'll say LAC-I is constantly being transcribed. So we'll say this LAC-I protein is like always just floating around in the cell, and it can kind of bind anytime, right? And then, there's some terminology. The operator sequence here, where the repressor can bind, we sometimes call it LAC-O as well, if you see that term anywhere. All right, so to set the stage, E. coli, they follow what's called dioxic growth. So this means that this essentially means that they're picky eaters. So in the presence of so they need sugar for energy, but if there's two different sugar sources, they prefer one over the other. So the two that they kind of debate between are lactose and glucose. So if there's glucose in the environment, the E. coli they'll eat up all the glucose first before they move on to the lactose. So it's not like they just eat whatever is like given to them. They'll specifically only eat glucose first and then only eat lactose. So glucose is the preferred sugar here. Right, so essentially, since glucose is preferred, we don't want to express the lac operon constantly, like all the time. Because think about it, the lac operon, we only need the genes from the lac operon, right? They're breaking down lactose. We only need those genes to be activated whenever we have lactose in the cell. Because why would I want to make proteins that break down lactose when there's no lactose in the cell, right? That's just a waste of energy. So the lac operon is going to be the way in which these guys control when exactly to express the genes for lactose breakdown. So the cell only wants to express the lac operon whenever there's no available glucose, because if there's glucose, then you want to use that up first, right? Because it prefers glucose. Then second, there is available lactose. So we want lactose to be there in order for us to make genes that break down lactose, or make proteins that break down lactose. So let's look at how this mechanism works. So the lac operon, the default state, the normal state of the lac operon is what we call bound or off. And this means the lac-I repressor that we talked about here, the lac-I repressor, it's normally on the operator. This means that the lac operon normally can't be transcribed and that the genes aren't being expressed. So as you can see here, the RNA polymerase can bind, but then it can't move forward because this lac-I repressor is here. Therefore, we call the lac operon an inducible operon because normally it's off, but it can be turned on by external factors. So it can be induced by other things. All right, so let's look at a couple scenarios. So first, scenario one, some lactose appears in the environment. So now lactose is here, so we want to express the genes to break down lactose, right? So what happens is this lac repressor is here. Lactose can actually bind. The lactose is the specific, what we call ligand, the specific thing that binds to the repressor. And once it binds to the repressor, it'll change the conformation of the repressor. So what happens is, as you can see on the right, first we have lactose. This lactose goes and binds to the lac-I repressor. And then what happens is that it changes the repressor shape. Now the repressor can't bind to the operator as well anymore, so it just falls off. And now, since the repressor is off, this RNA polymerase can move along and transcribe these genes. Right? And then remember, the operon codes for genes that help break down lactose. So this is perfect because this means that the operon can only be expressed whenever lactose is in the environment. So this lactose needs to bind. So yeah, it's like a sensor. If the lactose is there, it'll bind, and then the RNA polymerase can transcribe here. Now, scenario two. Let's say you have all this lactose that came to the cell. Now, all these beta-galactosidase, all these enzymes are created, and then they break down all the lactose, and you're out of lactose. You ate up all of it, and there's no more left. So now, as you can see, I represented that with all these X's here. So even the lactose that is bound to the lac-I repressor will get broken down, right? So whenever that happens, this lac-I repressor will return to its normal shape because there's no lactose bound to it anymore. And that means once it's back to its normal shape, it'll just return exactly back where it started. So the repressor is back, returns to its home on the operator. And now, again, the transcription here is blocked physically. So now the operon can't be expressed here. And again, this is perfect because if there's no lactose in the cell, then we don't want the lac genes to be expressed because it'll be a waste of energy. So this repressor binding is perfect. So review, no lactose means the repressor is bound, which means no transcription can occur. And then if there is lactose, then the repressor will unbind, and that means that transcription can occur. So this is really a fascinating system of self-regulation. And it's incredibly useful because it makes sure that bacteria aren't wasting energy when they don't need to. And it's incredible because there's no external, right? There's no enzymes that are coming in and actively like doing stuff. Like it's literally the system just functions on its own. It's just like, yeah, it's just really fascinating. So here's another layer of complexity to the operon. And this, I don't know if it'll be on your exams, but we're going to cover it just in case because it's a pretty important concept. So the lac operon, it also has this site upstream of the promoter called the cap site, or the catabolite activator protein site. So this cap site, there's this specific cap protein that can bind to this cap site. And when it binds, it basically increases the rate, so it increases the RNA polymerase coming onto the promoter. So basically when cap is bound, it increases the rate of transcription of the operon. All right, so whenever glucose, so let's say glucose, there's not that much glucose in the environment, and remember glucose is the preferred sugar for E. coli. So when glucose levels are low, a signal molecule called cyclic AMP or CAMP, this guy will start building up in the cell as glucose drops. And what happens is that CAMP is a specific thing that can bind to the cap protein. So scenario 1, we have low glucose. So remember, low glucose means CAMP is high. And what CAMP does is it binds to the cap protein, and then that causes the cap protein to bind to this cap site here. And remember, I said that when cap is bound to the cap site, this rate of transcription will increase. So more and more of these lac genes are being expressed when the cap is bound. Scenario 2, we have high glucose. So when glucose is high, the CAMP concentration is going to drop. Okay, so that means little to no CAMP is going to bind to this cap. So since there's CAMP isn't on the cap can't bind to the cap site, right? And this means that we're not promoting transcription at all, so there's little to no transcription here. So review, low glucose means more CAMP, means more cap bound, which means more transcription. And then we have high glucose, which means less CAMP, which means less cap binding, and then that means less transcription. So yeah, so why might this be a beneficial system for the cell? So when there's less glucose here, it leads to more transcription of the lac operon. So basically, the E. coli is saying, oh, the glucose, my favorite sugar, there's not a lot of it. So let me express more lac genes because I know lactose is there. So I want to, since I'm about to run out of glucose, I want to get more lactose genes so that I can start breaking down lactose for energy as well. So that's the rationale there. Then when there's a lot of glucose, right, the E. coli is like, oh my gosh, my favorite food is right there. So I don't need to make genes to break down lactose because I don't really like it that much. So I'm going to eat up all this glucose first and I don't need to express the lac genes, right? So essentially there's two signals coming from the lac operon here. The first signal is from the cap site based on glucose concentration. Then the second signal is coming from the operator. So the repressor being bound or not based off of the lactose concentration. So essentially the lac operon, it can respond to both glucose levels and lactose levels. So this gives more complexity to this entire circuit, and it lets the lac operon react accordingly, more specifically to the environment around it. So like I said, there's two essentially competing signals here. So excuse me, let's look on the right here. So basically, since we have either low glucose, high glucose, and then high lactose, low lactose, there's four possible scenarios that we can make. So I would probably put this on your cheat sheet maybe, but again, I don't know if the cap stuff is going to be on there, but it's good to know just in case. So first, if we have low glucose, but lactose is there, then remember low glucose means that there's a lot of CAMP, which means that the cap protein is bound, right? Then if lactose is available, remember the lactose, if you have the repressor, the lactose is bound to it, so it's coming off of the operon. So that means we have the cap protein promoting transcription, we have the repressor that's not blocking at all, so we have strong expression here. Strong expression, because there's a lot of transcription. This is great because low glucose and lactose is high, so we need high levels of lac expression. Next, we have, let's say there's high glucose, but there's no lactose. So high glucose, remember, high glucose means that the CAMP concentration is low, which means that this cap site is not bound by the cap protein anymore. So there's no stimulation here, essentially. Then again, lactose is unavailable, so there's nothing bound to the repressor to make it come off of the operon, so the repressor is just sitting there in its default state. So literally nothing none of the RNA polymerase is going to move this way, so the lac genes aren't expressed. Which is perfect, because why would we need to express these genes when we have no lactose and a ton of glucose? All right, third, let's say we have low glucose, but the lactose, there's no lactose either. So remember, low glucose means that this camp concentration is going to be high, so the cap site is bound here. But since there's no lactose, there's nothing stopping the repressor from coming off, so the repressor is just sitting there always. So even if we try to push against this repressor and try to get the RNA polymerase this way, it's not going to happen because this repressor is just always bound. So the lac genes aren't expressed. Finally, if we have high glucose and lactose available, this is going to be like a competing signal here. So first, the high glucose means that we have low camp, so there's nothing bound to this site here, right? So there's no promotion of the transcription, but lactose is available, which means that the repressor here, right? The repressor, lactose is bound to it and it's fallen off. So the operon is available to be transcribed. But since we don't have the cap site promoting the transcription, there will be a very low level of gene expression because this RNA polymerase, right, it can sometimes come here, but it's not really being pushed along by the cap site that much. So it's not like a minimal level of gene expression here. So that's how those like two competing signals work together. And yeah, like I said, this probably on cheat sheet, you don't have to know every single detail, but I just thought it'd be nice to include all this cool, like how you can think of this whole thing as like a circuit pretty much. Right, so let's move on to the second operon that we'll discuss for microemission. This is the TRIP operon. So the TRIP operon, it contains these 5 genes, TRIP-A through TRIP-E, and these are genes that are essential for the biosynthesis, which means just creating just the creation of tryptophan. So these enzymes are going to help create the amino acid tryptophan. And tryptophan is an essential amino acid that it's in a lot of our proteins. And a funny fact, there's a lot of tryptophan in turkey, like turkey meat. So we don't have to look at these specific functions of these, just know that it helps create tryptophan for the cell. And like the lac operon, we have the promoter here, the P, we have the operator, then we have this TRIP-R repressor that's like kind of being constantly made outside of this operon somewhere else. So this TRIP repressor is always going to be there. And this is like the equivalent of the LAC-I gene that's like elsewhere in the DNA, like in the lac operon. All right, so the operon also contains what's called TRIP-L. So that's this specific, what's called an attenuator sequence. And we're not going to look at this in this lecture, but whenever we move on to like the state level, that's when we'll probably review this because it's like a very complex mechanism. So yeah, so the TRIP operon, it's like the opposite of the lac operon because the default state is on. So the default state is when this repressor is not bound to the operator, and then the RNA polymerase can just proceed and then transcribe, right? So this means that we call the TRIP operon a repressible operon because normally it's on, but we can repress it by some environmental factor, right? So the same, so a similar idea here. So the cell, we only want the operon to be on whenever we don't have tryptophan in the environment, right? Because if we already have tryptophan in the environment, then we don't need these genes to make even more tryptophan. We can just use the stuff that's already in the environment. So we want to turn the operon off whenever there's tryptophan. Then whenever there's no tryptophan in the environment, we want to turn this on so that we can make tryptophan, right? So let's look at the mechanism. Like I said, the TRIP repressor normally unbound, allowing transcription of the genes in the operon. So scenario 1, no tryptophan in the cell. And then this is the same logic by which, so remember how the lac repressor, we'll look at this in the next slide, so I won't mention that for now. So scenario 1, we have no tryptophan. The repressor is not bound normally, right? Because the operon is normally on. So this repressor is just sitting, is floating around, and then RNA polymerase can just come up and then transcribe and then make these enzymes that synthesize tryptophan. So this is when we have no tryptophan, right? Now, if we do have tryptophan, so now the opposite effect from the lac operon is going to happen. So remember, in the lac operon, we had lac I. Once a lactose molecule binded to it, it fell off, right? Like this, and then you could transcribe. In the trip operon, it's the opposite. So when the repressor is normally unbound, but whenever the tryptophan is in the environment and it binds to the repressor will actually go on this time. So essentially we have tryptophan in the environment, it binds to the repressor can now bind to the operator, and now it's again physically blocking this RNA polymerase from proceeding forward. Like this. And again, this is perfect because if we already have tryptophan in the cell, we don't need to make the tryptophan enzymes because it's already there.